ready to start the last lecture on that by Laurent. And Laurent is going to be looking at human infections in the body.
Yes, thank you. So for this lecture, I will focus on three main issues here. Climate change, ocean acidification, and ocean deoxygenation. So we talk about the impact of climate change on the first levels of the trophic fluids. We'll talk about changes in pH and ocean acidification, and the last part of the talk will be ocean deoxygenation. So I start with very few general slides. I start with these iconic figures, not maybe as famous as the clean curve that Raghu showed you this morning, but you have here the mean, or the average temperature, sea surface temperature, evolution from 1860 to 2000, reconstructed by several groups here, and you clearly see this warming of up to 1 degree over the past century. So we'll be looking at the impact of this warming and the changes in ocean dynamics that are partly due to the warming onto marine biochemistry and the lower trophic levels. The other big change in the ocean that is also due to increasing CO2, but that is not climate change per se, is ocean acidification. And you have here a record of surface of ocean pH at one specific location, the Bat Station, which is localized in the Sargasso Sea. And we have direct measurements of ocean pH from the beginning of the 80s here to today, and you clearly see, despite this strong variability, you clearly see this decreasing pH of the ocean surface waters as recorded here at Bats. Over the last 50 years, the ocean has lost maybe 0.1 pH units. And of course, this is due to the fact that the ocean is absorbing part of the anthropogenic CO2. So those are two big changes here on here, the ocean surface, warming and acidification. So this graphic here comes from the last IPCC report in 2007, and it's a schematic that shows you the different physical and chemical changes that are happening today in the ocean. You have a warmer ocean, here mostly in the tropical but also at mid and high latitudes. You have, of course, reduced sea ice extents. You have freshening of high and mid latitude waters, so changes in the salinity. And you have the chemical changes that I was mentioning. Decreasing pH, that's the yellow circles here. And you have also a showing of the saturation horizon for calcite and aragonite. We'll come back to that in the lecture where I will mention ocean acidification. So that's the chemical and physical changes in the ocean that are both due to increasing greenhouse gases in one case, that's the physical changes, and to only increasing CO2 in the other case, that's acidification. So there are other changes, large-scale changes, that will have impacts on marine biogeochemistry, changes in the nutrient delivery to the ocean. We have here two maps from a model work published in 2008 by Juice et al. That show how much the nutrient delivery of nitrogen to the ocean has changed over the past decades. Maybe multiplied by 4 or 5 from the natural nitrogen delivery to the ocean in 1860 to now almost 4 times higher numbers here. And of course, this difference is due to combustion and to agriculture that will put nitrogen in the atmosphere and that will be delivered through the ocean. So that's atmospheric deposition only, and you have, of course, another source of nutrient to the ocean, which are the rivers. And one clear impact of nutrient delivery, or increasing nutrient delivery by the rivers, is what has been here documented by Diaz and Rosenberg in 2008. It's an increasing number of coastal electrification sites and hypoxia. So they've looked and they've gathered here all the sites that have undergone electrification and hypoxia, and that's clearly linked to increasing nutrient delivery. So if we summarize all the basically major drivers that are changing ocean biogeochemical cycles today, you have here carbon dioxide directly that affect ocean chemistry and that will drive ocean acidification. Then carbon dioxide, but also the other greenhouse gases change the climate. That will, of course, alter primary production, for example, but all biogeochemical cycles. And then you have what I was just mentioning, increasing nutrient delivery by rivers, but also by atmospheric deposition here, that drive coastal eutrophication, but that may also change global scale primary production in the ocean. So in this lecture today, I will focus only on those topics here. I will show you how climate change may alter primary production in the ocean. Then I will focus on acidification and show you how the models project changes in acidification and ocean chemistry in the next decades. And I will also mention in the last part of the talk quite a new subject, deoxygenation, that can be caused both by climate change, changes in circulation and warming of the ocean, but also by acidification. And to do that, I will make use of the recent simulations by the IPCC class models that have been run for the upcoming IPCC reports, IPCC AR5, that will be out in 2013, so quite soon. So all those simulations have been done. All the data, the output of the models simulations have been gathered. And lots of groups now are looking at those simulations. And something that is quite new from the previous IPCC exercise is that now many of the recent IPCC class models do include some marine biogeochemical components. The reason for that is not marine ecosystem studies, it's mostly because of the carbon cycle and climate linkages, but we can also look at those simulations to study biogeochemistry in general and ecosystems. Just one word on the scenarios that are used in these new simulations for the next IPCC reports. Maybe some of you have been used to the stress scenarios that have been used for the past two reports, 2007 and 2001. And now we move to those new scenarios that are representative concentration characteristics, that's 3 letters. And you have numbers here that basically tell you how many watts per meter square in addition you have at the peak of the change. So 8.5 means that in 2100 you have plus 8.5 watt per meters because of anthropogenic perturbations. And you can see here only one of the drivers, CO2 concentrations, in the 4 scenarios, 8.5, 6, 4.5, and 2.6. So the 8.5 is quite a pessimistic scenario if you want, with the atmospheric CO2 up to 900 ppm. And the 2.6 is a peak and decline scenario in which atmospheric CO2 doesn't reach more than 450 ppm. So I will mostly, to illustrate the results of my talk, use the RCP 8.5. That's basically the easy way to look at the mechanisms simulated by the models. But of course, we have simulations with all those scenarios here. So outline of my talk, a few words on the marine biogeochemical models that are embedded in Earth system models, in climate models. Are they different from what we heard about this morning, for example, or yesterday? I will show the evolution of those models that are embedded in Earth system models over the past few or 2 decades. And then I will say a few words on evaluation of those models and explain why it's more complex to evaluate marine biogeochemical models embedded in Earth system models than marine biogeochemical models that are forced by observed analysis, for example. Then that's the core of my lecture here. I will say a few words about the changes in NPP driven by climate change and how this is simulated by the CEMIC5 models. So CEMIC5, that's the comparison exercise, so couple model into comparison project that is used by IPCC Layer 5 to basically diagnose and project how climate will change under human perturbations. So changes in net current productivity driven by climate change, then I will focus on changes in pH and changes in oxygen. And I will mention some issues or challenges for the future. So in fact, the first models that have been used and embedded in climate models were very simple geochemical models. We cannot call them biogeochemical models if you want. In fact, the only biological equation that was embedded in these models is the following. The only flux, if you want, of interest was the flux of particles towards the deep ocean here, crossing the euphotic layer at 100 meters. And that's basically the flux that is important for RC CO2 fluxes on many timescales. So no matter or no interest in simulating explicitly phytoplankton zooplankton here. And in those models that were developed in the 80s, beginning of the 90s, the export production was computed as a function of temperature, of light, and of one nutrient, usually phosphate here, but it could be also nitrate. And those geochemical models were integrated in ocean general circulation models and were used in climate models, mostly to predict and compute RC CO2 fluxes here that would drive or would change atmospheric CO2. Then in the 90s, most of the climate groups, or some of the climate groups, did integrate a bit more complex geochemical models by representing explicitly phytoplankton, zooplankton, some particles dissolved organic matter here. What we heard about this morning, MPZD, and yesterday also, is you heard about MPZD, and you'll be working on MPZD models this afternoon with Rebecca. So in that case, most of the models still very simple with one nutrient, only one very generic phytoplankton, one generic zooplankton, dissolved particles. Maybe just illustration, I don't know if you've seen that yesterday. You could ask the reasons why we think we can represent all phytoplankton species by only one big generic group, and there are some good arguments to say that this is possible at some extent, and one of these arguments comes from the work of Epley in 1972. You have here the growth rate as a function of temperature for several individual species here of phytoplankton, and you see some cold species like this one, and some, of course, warmer species like this one, and Epley drew this curve here, that's an envelope curve, and basically that's what we use in the models to represent the influence of temperature on phytoplankton. So that's the way you can gather information you have on very different species to basically extract some general, maybe, relation between temperature and growth rate. So maybe it's easy for temperature, it may be more difficult for nutrients and for some other functions, but there is some ways to derive parameterizations for generic phytoplankton. Of course, this was too simple for most of the things that we are interested in now. And so the new generation of models, and some of the models that are now used by climate groups and in climate models, are dynamic, what we call dynamic green ocean models. And the phytoplankton group here has been split in different phytoplankton functional groups by size or by functions. We have also several zooplankton, and maybe the most important here, several nutrients, because we know it's not always the same nutrient limiting in the same regions of the ocean. And one big addition from phosphate or nitrate to this new generation of models is the addition of iron limitation here. That is very important in many regions of the world. But apart from that, the philosophy between those two different models is very, very similar. So you've heard about the BDC model yesterday, I guess, in the lecture by Marcus. And we showed just a few evaluation results from this model, which is the Pisces model, which is very similar to the models you've been described, with two phytoplankton groups here and two zooplankton groups, and the 5 main nutrients here. And so those models, so that's just the biogeochemical part, but of course they are embedded in an ocean general circulation models. And as Barish was explaining this morning, we have these equations, and that's chlorophyll at the surface of the ocean, and at each time step you compute the evolution of the total chlorophyll you have in each of the cells of your model here, based on advection, diffusion, and of course the biogeochemical processes. But now, and so what you can do is of course compare here surface chlorophyll from satellite observations to your model outputs. That's the first way you start to evaluate your model. You see that now we try to go, of course, a little bit further than that. That's, again, the Pisces model embedded in the ITSM climate model. So this time you have it in a climate model and you compare to observations. Maybe I want to mention this. So in the evaluation procedure, the first task is, of course, to compare to climatologies. And in that case, you will focus on mean state and seasonal cycle of chlorophyll, of course, but also nutrient, oxygen, some other variables. And the big question is this evaluation, is this relevant for century-scale projections? If your model performs very well for the seasonal cycle, does this mean that it will be better for projections? So basically, we try to find other ways to evaluate the models on longer timescales, and maybe timescales relevant to what we have interest in. So the second step here is of course to use internal to decouple validity.